There's more than one way to make synthetic human insulin, but in this video I'm going to focus on the traditional method, a method that involves synthesizing the A chain and the B chain of insulin separately in two separate strains of E. coli bacteria, making fusion proteins with beta-galactosidase, and then cleaving those and combining the two peptides, the A and B chains, to make a final functional human insulin protein. It's the technique that was used by Genentech way back in the late 1970s and the early 1980s. It's the original technique for making human insulin, and it's still widely used today. As far as proteins go, insulin is a very small one. It has just 51 amino acids. Now, to put that into context, another protein that we'll be looking at in this video, beta-galactosidase, has more than 1,000 amino acids, but insulin has just 51. But it does have quaternary structure. That is, it's made of two separate peptide chains. And by peptide, I mean a short polypeptide. We wouldn't normally use the term polypeptide unless it had more than 50 amino acids. And both of the chains in insulin are shorter than that. In fact, the short chain, the A chain, has just 21 amino acids. The B chain has 30, and they're held together by two disulfide bonds between cysteine amino acid residues. As I mentioned before, the approach taken by Genentech in the late 1970s was to synthesize the A chain and the B chain separately in two separate strains, different strains of E. coli bacteria. And you may think, well, that's a strange thing to do. But one of the main reasons that they did that was because there were great concerns that E. coli, E. coli is a bacteria that causes gastrointestinal diseases in humans, and there was great concern that genetically modifying E. coli so that it produces a human protein may have untold terrible consequences. As it turns out, those fears were unfounded because the particular strain of E. coli that was used, called K12, isn't known to cause any human diseases at all. It's a non-pathogenic strain of bacteria. So, the fears were unfounded. Nevertheless, that was the main reason for doing this. The idea was we would synthesize the A chain in one bacteria, the B chain in another strain of bacteria. Then only after we've taken those proteins out of the cell would they be combined to become a functional insulin protein. But it would never be functional in any of the bacteria at all. Okay, now let's look at the way that the A chain of insulin is synthesized. And of course, the B chain is much the same thing, but we'll just focus on the A chain and you'll get the idea. The first step is to get a gene for the A chain of insulin. The only complication with that is you can't just cut it out of a human cell because the genes in human cells have introns in them, and bacterial chromosomes, bacterial DNA, doesn't have any introns. Bacteria don't understand introns. So if you just take a human gene with all of its introns and you put it into a bacteria, that won't work. We need to get a copy of the insulin A gene without introns. And there are two ways that we can do that. The first way, which is the way that people would typically do it today, is to use what's called cDNA or copy DNA. And the way you get that is by, first of all, isolating the messenger RNA molecule that encodes insulin A, and then using an enzyme called reverse transcriptase to transcribe it backwards into DNA. All right. Now, that works, of course, because messenger RNA doesn't have any introns in it. Okay, the introns get spliced out of the primary transcript, the pre-messenger RNA, to make that messenger RNA. So if you get messenger RNA and transcribe it backwards, the introns are gone. So that's one way of getting the gene for insulin A without introns. And as I said, that's probably the way that it's mostly done today. But back in 1978, when Genentech was doing this, they didn't even know where the gene for human insulin was. They didn't even know what the sequence of bases was or anything. All they knew was the sequence of amino acids in the insulin protein, in the A chain of the insulin protein. So because they knew the sequence of amino acids, what they did was they made a synthetic DNA molecule in a laboratory, right? They put it together nucleotide by nucleotide to encode the same amino acid sequence. Now, as it turns out, the sequence of bases in the gene that they made is completely different to the sequence of bases in the natural human insulin A gene. But that doesn't really matter because as long as it encodes the same amino acids, we end up with the same protein, right? And the idea here is that the genetic code is degenerate or redundant, right? I mean, CUC codes for leucine, but so does CUG and CUA. In fact, CU anything codes for leucine. So it doesn't really matter which codon you use. As long as it codes for leucine, you're still going to end up with a leucine at that position in the protein. And so that's the approach that Gen took. Either way, we end up with a gene that encodes insulin A. Now that gene needs to be prepared with a couple of restriction sites, okay? And those restriction sites, so we've got some sticky ends here. One of those is created by the restriction enzyme EcoR1, which you can see has this restriction site here. It's cut the DNA there. The other is BAMH1, which cuts, again, leaving sticky ends, but they're different sticky ends to EcoR1. All right, two different sticky ends on either side of the insulin A gene. And then we need to get a plasmid. Now, the plasmid that they used was called PBR322, not that that's particularly important, but what's interesting about it is that plasmid has two genes for antibiotic resistance. It has one gene for ampicillin resistance and one gene for tetracycline resistance. But the restriction site for BAMH1 is in the middle of that tetracycline resistance gene. The other one's just over here on the plasmid somewhere. But what's going to happen is when the plasmids are mixed with those two restriction enzymes, both of those sites will be cut by each of those restriction enzymes. And what we hope is that a chunk of the plasmid is going to get cut out of there, and that the human insulin A gene will take its place and stick in there, because the sticky ends on either side of that human insulin gene are complementary to the two sticky ends cut in the tetracycline gene and that other site on the plasmid. Of course, if that happens, that tetracycline gene is now no longer a fully functioning tetracycline resistance gene, because half of it is missing. So you'll notice that we no longer label it tetracycline R for resistance, it's now tetracycline S for susceptibility, because a bacteria that has this plasmid in it will not be resistant to tetracycline like it would have been if it had that previous plasmid. So when we look now at the possibilities, these are the potential options, right? Some of the plasmids won't have been modified by these restriction enzymes. The restriction enzyme might have cut this open and it just rejoined, and cut this open and it rejoined. So we've still got a plasmid, but it's not a recombinant plasmid. It doesn't have the human insulin A gene in it. On the other hand, we'll have some plasmids that do have the human insulin A gene, and the real difference between them, remember, is that these ones are going to be, well, they've got broken tetracycline resistance genes, so bacteria with these plasmids are going to still be tetracycline susceptible, whereas a bacteria that takes up this plasmid will be resistant to both antibiotics. So here we have an E. coli bacteria that's taken up one of these plasmids, another E. coli bacteria that's taken up one of these plasmids. Of course, to do this, the plasmids and the bacteria are mixed together and treated with, either chemically or electrically or with heat or something. In some way, there's various different ways to make bacteria competent to take up plasmids. So we're going to end up with 3 different kinds of E. coli bacteria in the mixture, aren't we? We're going to have some bacteria that don't take up any plasmids at all. We're going to have some bacteria that take up a plasmid that's just a normal plasmid. They've been transformed, but not by a recombinant plasmid. And then we'll have some E. coli that have been transformed by a recombinant plasmid. The trick, of course, is to identify the ones that have taken up a recombinant plasmid. You might be thinking to yourself, yeah, surely there's another combination as well. What happens if a plasmid gets cut open and this chunk gets cut out, but then the plasmid just joins back up again? All right. Well, that won't happen, of course, because remember, these sticky ends are not complementary to those sticky ends because they're cut by 2 different restriction enzymes. So the only combinations are these ones. And again, our challenge now is to identify the bacteria that have taken up a recombinant plasmid. So how do we do that? Well, what we do, first of all, is grow the bacteria, culture the bacteria on an agar plate that contains the antibiotic ampicillin. Now remember, one of the genes on that plasmid gives bacteria resistance to that antibiotic. So any bacteria that grow on this plate must be resistant to ampicillin. Any of the bacteria that didn't take up a plasmid at all won't grow on this plate because the ampicillin will prevent them from growing there in the first place. So if a bacteria has grown here, we know that it's taken up a plasmid. But that doesn't tell us whether it's taken up a recombinant plasmid with the human insulin A gene in it, or just a regular plasmid that doesn't have the human insulin A gene in it. So how do we find out which ones have taken up which kind of plasmid? Okay, they've all taken up a plasmid. How do we know which ones? Well, we get another agar plate, but this one has tetracycline in it, that other antibiotic. Right? Now remember that the bacteria that have taken up a recombinant plasmid, that recombinant plasmid with the human insulin A gene in it, they are no longer resistant to tetracycline. So if we plate these across, we scrape some of this off and put it in here, we scrape some of this one off and put it in here, we scrape some of this one off and put it in here, and some of this one off and put it in here. And you'll see that colonies have grown in each of those places except for here. So what that tells us is that this culture here contains the recombinant plasmid, because when we transferred them across onto this agar plate, they couldn't grow. They've got a broken tetracycline resistance gene. So we go, in other words, if we look at the plasmids, that's what they look like, right? So this colony here wasn't able to grow there because its tetracycline resistance gene has been interrupted by the human insulin A chain gene. Okay, so what we do now is we come back to this first plate and we just scrape off some more of those and grow a culture out of those. Okay, so we just come back to them. As long as we remember where they came from, we can just go back and get some more of them, because we know that colony now contains the recombinant plasmid. The next step then is to get some of those bacteria and to purify the plasmids out of them. Okay, so the plasmids, the bacteria are lysed, the plasmids come out, and they get purified using various laboratory techniques so that we get a whole bunch of these plasmids, because they're the plasmids that we want. All right, so we've got a whole bunch of plasmids that are the kind that we want. Then what we do is a second step. What we do is we get that same restriction enzyme again, echo R1. We're not going to use the BAM H1 again this time. We're just going to use echo R1. And that will cut the plasmid open here at that same place, because that restriction site is still there, of course. It's going to cut the plasmid open like this. Now, that's the first thing. The next thing that we want to do is get another piece of DNA which contains the gene for beta-galactosidase. Now, beta-galactosidase is a protein that we'll talk about in a moment, but it's basically it's an enzyme that breaks down lactose into glucose and galactose. All right, and it's on this piece of DNA, there are three parts. There's the gene that we call LAC-Z, or LAC-Z in Australia, but the LAC-Z gene, which encodes the protein itself. Then there's a promoter and an operator. Okay, so this is just like at the beginning of an operon. In fact, the LAC-Z gene comes from an operon. It's one of the genes from the LAC operon. All right, so we take this, but it's been cut using echo R1, the same restriction enzyme that we've used to cut open the plasmid here. And so what we're hoping is that beta-galactosidase, that's easy for me to say, the beta-galactosidase gene will join into the plasmid like that, forming another recombination of this plasmid. All right, so it's now a recombinant plasmid because it contains the human insulin A gene, and it also is recombinant because it contains the beta-galactosidase gene as well. So once again, our challenge is to mix these with bacteria and encourage those bacteria to take up plasmids, to make the bacteria competent to take up plasmids. And again, of course, there are three combinations, three different types of bacteria in regard to this. Some of the bacteria won't take up a plasmid. Some of them will take up one of these plasmids, and some of them will take up this plasmid that has not only the human insulin A gene, but also the beta-galactosidase gene as well. And that's the one that we want to get hold of. So again, we need to figure out how we can find that bacteria. Of course, we can't use tetracycline this time because both of these plasmids have broken or interrupted tetracycline genes. So this bacteria and this bacteria, both of them will be susceptible to tetracycline. So we can't use tetracycline anymore to differentiate between these two. In fact, the only difference now between these two is this beta-galactosidase gene. So how do we use that to find the bacteria that have taken up that plasmid? Well, beta-galactosidase is an enzyme that breaks lactose, it hydrolyzes lactose into glucose and galactose. That's its normal job. But in a laboratory, scientists have found that there's another molecule called, with a very sexy and memorable name, 5-bromo-4-chloro-3-indole beta D-galactopyranosidae. You can't even say it. Such a big word. You can understand why, I made it almost to the end of that word, but you can understand why people prefer just to call it X-gal. It's a much cooler sounding name. Anyway, X-gal is kind of a little bit like lactose. It's made of a galactose together with this other molecule that contains an indole. And ordinarily, X-gal is white in color. It's a white color. But beta-galactosidase, that enzyme, will not only cut lactose into glucose and galactose, but if it finds an X-gal molecule, it will cut the galactose off that too. And when it does, the indole part goes bright indigo blue. All right, so that's really cool because that means if there's a functioning beta-galactosidase gene in a cell, in the presence of X-gal, we're going to see the color blue. So we put those on a plate that contains ampicillin and X-gal. Now, of course, the ampicillin will prevent any bacteria from growing there that don't have a plasmid in them. And you might think, well, what does it even matter? Because they're not going to go blue unless they've got, the beta-galactosidase gene. And so we don't even care about all the other ones. But I guess this also prevents contamination of the plate as well. It means that, bacteria that are just, if you ever made agar plates, you take the lid off there, you're likely to get contamination on there. So having ampicillin in there prevents that from happening. And besides, all these bacteria are resistant to ampicillin, so why not? It's not going to affect them, but it will prevent contamination. But it's the X-gal here that's really important because those colonies that have turned blue, the reason they've turned blue is because they have the beta-galactosidase gene in the plasmid that they've taken up. The colonies that have stayed white, they haven't taken up a beta-galactosidase gene, so they can't produce beta-galactosidase, so they can't cut X-gal into galactose and that blue indole molecule. So what we need to do is now take some bacteria from one of those blue colonies and get the, because we know that those blue colonies there now, those are the bacteria that have taken up our recombinant plasmid containing the human insulin gene and the beta-galactosidase gene. And the importance of that we're going to talk about in just a moment. But first, I just want to show you what this looks like in real life, because obviously this is just my drawing. And there's a photograph of what it actually looks like. And you can see clearly some of those colonies have gone a beautiful indigo blue and others are still white or clear. We haven't yet answered the question why we really want that LAC-Z gene there in the first place. Why don't we just want, because both of the plasmids both have the human insulin A chain gene in them, right? So whichever bacteria take up that, they've got the human gene in it. Why do we need the human gene right there beside the LAC-Z gene or the beta-galactosidase gene? Why do we need it there in the first place? Well, the reason is that we need to make a fusion protein. And we'll talk about why that is in just a moment. But first of all, let's talk about what it is. A fusion protein is a protein made of several proteins that are still fused together. Ordinarily, at the end of any protein, there's a stop codon. But in the production of this LAC-Z gene that we put in the plasmid, the last 17 codons are cut off. Okay, so I mean, remember, because beta-galactosidase, remember, is a huge protein, massive. There's only 17 amino acids missing at the end of it. That's not very much. It's not enough to stop it functioning as a beta-galactosidase. 17 codons at the end isn't enough to stop it from being a functioning beta-galactosidase. But it does mean that the stop codon is missing. And because there's no stop codon, it means that when a ribosome translates the messenger RNA produced from this gene, it's not going to stop and turn that into a protein and then start again and turn that into a separate protein. It will make it all as a single protein. All right, but before we even get to that, notice that between the promoter and the LAC-Z gene, which for beta-galactosidase, there's a protein called a repressor, and that repressor stops the gene from being transcribed. Okay, this is gene regulation. It's a bit like the TRP operon. It's another operon called the LAC operon. The LAC-Z gene is the first of the genes in the LAC operon, naturally. And it's prevented from working by this repressor stuck on the operator. One of the reasons why we want the LAC-Z gene as a fusion protein with insulin A is because it gives us the ability to turn the gene on and turn the gene off at will very easily. Because to turn the gene on, in other words, to unblock this gene, to unlock it, all we need to do is add some lactose, or another compound called IPTG, which is a synthetic kind of analog for lactose. Either way, a scientist can simply put one of those into the mixture, into the cell, and that lactose, or allolactose it would need to be, will bind to the repressor, change its shape, and once the repressor changes shape, it'll come away from the operator. The RNA polymerase can then go ahead and transcribe those genes into messenger RNA, and then a ribosome will translate them. But again, because there's no stop codon at the end of the LAC-Z gene, it makes one single protein, one single polypeptide chain that is sort of the fusion of both beta-galactosidase and the human insulin A gene. This is what that looks like. Well, this is actually insulin B together with beta-galactosidase, but the same thing. It's this massive beta-galactosidase polypeptide, remember 1,000 amino acids, and then the little insulin peptide tacked onto the end of it as a fusion protein. And you might think, well, what's so good about that? Well, it turns out actually that it's really important that we do it this way, because in E. coli, there are enzymes, proteolytic enzymes, that will very rapidly degrade any short unfolded peptides. Okay? If there's a short unfolded peptide, these enzymes will break it down really quickly, but they don't do that to big proteins. And so by having the insulin peptide attached to this great big beta-galactosidase protein, it sort of protects it, sort of like a parent holding the hand of a child. It kind of keeps it safe. These enzymes won't attack that insulin peptide while it's connected to the beta-galactosidase protein. And so, that's the main reason we want to do this, right? Because you could just put a promoter on the front of the insulin A gene, right? And make these insulin A peptides all by themselves, but they'd just be rapidly degraded by the E. coli cell as they were produced. This prevents that from happening. This is a summary of what we've just been looking at. Here, of course, is our plasmid with these two important genes, the beta-galactosidase gene, followed immediately by the insulin A chain gene. And as we've already said, the last 17 codons of the beta-galactosidase gene are missing. And most importantly, the stop codon is missing, which means that it all gets transcribed and translated as a single polypeptide chain, giving us this fusion protein that has both the beta-galactosidase and insulin A. But what's this green dot here? Well, that's the last thing that I want to point out here, or second to last actually, and that is that at the amino end of the amino acid chain, that is the N-terminal, there's an extra codon for methionine that's added when the insulin A gene is being synthesized. there's an extra ATG that is on the coding strand. Of course, on the template strand, that would be TAC, but that codon for methionine, is added at the N terminal, the nitrogen terminal of insulin A. And this is true of insulin B as well. And why is that? Well, it turns out that there is no methionine at all in either of the chains of insulin. There's no methionine in there at all. So by adding that methionine, it means that there's an amino acid joining the beta-galactosidase to the insulin peptide that doesn't exist anywhere else in the insulin peptide. And that means it can be chemically treated to break that amino acid joining them together. And there's a compound called cyanogen bromide. So once our fusion protein is made, held together by this methionine amino acid here, the proteins are extracted from the bacteria. The bacteria needs to be lysed and the proteins are extracted from that. But then they can be treated with cyanogen bromide to release the insulin A peptide from the beta-galactosidase protein. And that's how insulin A and insulin B, for that matter, are produced. The last step of the process is actually the easy part, and that is combining the two chains, the A chain and the B chain, together to make a single human insulin or humulin protein. Humulin's the name that Genentech sold it under, or Eli Lilly, which is the company that took it over. But this is the easy part, because it turns out that when the A chain and the B chain are combined in vitro in a laboratory, they just naturally, spontaneously form insulin. That is, the disulfide bonds between the two chains form spontaneously. They fold up spontaneously into the shape that they need to be to become human insulin, exactly the same as the human insulin that's produced by our own pancreas. Isn't that fantastic? And it's amazing to me that, this company, Genentech, the company was just formed in 1977, and just 4 years later, they had perfected this technique enough that they were able to market and sell human insulin to diabetics. And I think in 4 years, that's a remarkable achievement.